Hi everyone, my name is Janki Patel with today's sponsor Agilex Biolabs, and I will be moderating today's webinar hosted by Endpoints News, Solving Complex Immunogenicity Challenges. Thank you to Endpoints News for hosting us, and I'm so excited to introduce our panel for today. Joining us, we have Agata Mikalik, the Research and Development Manager, Immunoassay at Agilex Biolabs. Also joining us is Morgan Evans, the Principal Method Development Scientist, Immunoassay at Agilex Biolabs, and also Emmeline Laruto, the Senior Method Development Scientist, Immunoassay at Agilex Biolabs. Now, if you do have any questions during today's webinar, our panelists have reserved some time at the end. So please hit the Q&A button at the bottom of your screen as soon as you think of it. This webinar will also be available on demand tomorrow and to rewatch and to share with your colleagues. So without any further ado, I would like to hand over to Agata to start our today's session.
Hello, everybody. My name is Agata Michalak, and I work as an R&D manager for the Immunoassay group. My group is responsible for developing ligand-binding assays to support clinical and preclinical studies. Previously to joining Agilex, which was 5 years ago, I was in similar roles in Europe, and I have over 18 years of experience in bioanalytical and diagnostic industry. And for the past 14 years, I have mainly focused on bioanalytical method development, specifically for large molecules, and I have a master's degree in biotechnology.
Morning, everyone. My name is Morgan Evans. I'm a principal scientist here at Agilex Biolabs. So my primary focus is the development of pharmacokinetic immunogenicity and biomarker assays. Just like Agata, prior to Agilex, I was working over in Europe for major CROs as well as in academia, and I also had a slight focus there in the biomarker discovery world. So thanks for joining today, and I hope you gain something from this presentation.
Hi everyone, my name is Emmeline Laruto. I work as a senior method development scientist at Agilex. I have been working in immunology for over 8 years. I first started in vaccine development, and then I gained expertise in PK, ATA, and biomarker assays. I have most experience in developing and validating immunoassays that meet clients' requirements under GLP regulations. So at Agilex, I'm responsible for developing immunoassay for the support of preclinical and clinical studies. Now let's jump into the webinar. I'll hand over to Agata.
Thanks, Emmeline. In today's presentation, we're going to give you a brief overview of a few aspects of immunogenicity testing, including its criticality in drug development process. We'll discuss the key parameters for those methods and current challenges associated with development of those methods. We'll talk about Agilex capabilities. But the main part of the presentation will discuss 3 unique case studies that will highlight the importance of tailoring assay development process to both the drug molecule and also the clinical study. And we'll present a few tools to assist you in tackling drug and target tolerance in those assays.
So just as a brief introduction to immunogenicity, as some people may be aware, immunogenicity is the ability of an antigen to elicit an immune response within a patient or person of interest. So here on the slide, we can see a really basic schematic of a B cell antibody response, where we have an antigen binding to our B cell, and through some downstream differentiation, antibodies being secreted. What's important in the drug discovery world is what the antigen actually is. And when we talk about immunogenicity, we're talking about the drug or the therapeutic of interest being that antigen. So what's really important to consider is that drugs can come in 2 forms of immunogenicity. So we have wanted and unwanted. Wanted is typically for our vaccines. So as we're all quite aware now, having a COVID-19 vaccines, we really want to see a big immune response to that vaccine so that we can be sure that when we come into contact with that pathogen of interest, that we do have the immune response there to protect us. On the flip side of that, and what we're really going to focus on today, is unwanted immunogenicity. And when we talk about this, really we're talking about other biotherapeutics such as monoclonal antibodies, where those immune responses may actually elicit some downstream effects that we don't want to see in our patients or volunteers during our trials.
So immunogenicity testing plays a critical role in drug development process. Patient safety is obviously the primary concern here, because the presence of ADAs can cause adverse responses like allergic or anaphylactic reaction, or even induction of autoimmunity. However, safety is not the only concern. Accurate measurement and modeling of PK and PD profiles is required to make key decisions regarding dose levels and dosing frequency. The presence of ADAs can impact those profiles. They can be impacted in diverse ways. Presence of ADAs can enhance clearance, but it can also sustain the circulation of therapeutic protein. And generally, ADAs that are present in high concentration and are of high affinity have the highest potential to impact those profiles. And ADAs can modulate and even neutralize the drug therapeutic effects. That's why it's critical to assess immunogenicity in clinical drug development.
So here we can see some key parameters that we really want to be focusing on as we do any assay development during immunogenicity testing. So first we have the sensitivity of the assay. So here we want to be looking at an acceptance criteria of being able to detect any anti-drug antibodies down to a level of less than 100 nanogram per mil. So this is to make sure that we can detect all clinically relevant anti-drug antibodies, and is actually now a regulatory requirement as well. But it's important to consider that this is based on the positive control within the assay, which may or may not be truly reflective of your patient samples. Further, we have the drug tolerance of the assay. So here, this is about how much free drug is actually available in each of those time points that are being collected from patients. And here we want it to be clinically relevant. So by that, I mean, we need to optimize our assay to be able to cope with having the amount of drug that is given in any of those time points. As well as that, we have the target tolerance. So here I've written that it needs to be biologically relevant, and this is really coming into play, especially as we move throughout populations. So as we move through trials and maybe move from a healthy to a diseased population, where the target of interest may go up or down, this can be quite difficult to make sure that our assay stays in a fit for purpose way. And this is really difficult when we talk about novel targets, as we may not fully understand the mode of action. So at Agilex, we tend to use a standard assay, which is commonly known as the homogeneous bridging assay for most of our developments where we can. And this is here on the left-handmost pane. So within this assay, what we do is we actually coat our wells with a biotinylated version of the drug, sticking to a streptavidin plate, which is essentially the biological superglue of the world. So we know that this isn't going to come away. Any anti-drug antibodies present in the samples will then bind to that drug on the plate, and subsequently we detect it with a sulfur tag labeled drug, which when stimulated produces a light signal. So when we start to talk about our target interference, you can see here that there's two examples on the screen. So first we have multimeric targets. So multimeric targets are targets that have more than one binding site available for the drug. And when that occurs, it actually means that they can cause the same bridge that the anti-drug antibodies have actually been causing in the assay. And when that happens, we can run the risk of false positives. On the flip side of that, we have monomeric targets. So these are targets that may only have one binding site available to the drug of interest. And here you can see in this right-handmost pane that they actually block the bridge being formed. So here we run the issue of false negatives within the assay. As well as that, we have the drug tolerance or drug interference within the assay. So again, this is where there's a large amount, or potentially a large amount, of free drug available in the patient's serum samples. And what they're essentially doing is mopping up all of the available anti-drug antibodies and preventing those to binding to our assay reagents, which of course are based on the drug itself. So again, we're running the risk of false negatives if we don't adequately optimize our assays.
So now let's look at the challenges that are being faced when developing immunogenicity assays. So typically, direct formats were being used, but as we move to MSD platform, where the drug can be used as capture and detection, this has reduced the need for alternative reagents. So reagent availability and platform technology of choice are more considered historical challenges. And then the interaction between the drug target and sample is really important to consider, especially as new complex modalities are being developed. So if we look at fusion protein, bispecific antibodies, and antibody drug conjugate, for instance, it means the interaction can be more complex, and then there's a need for more complicated assay formats. And then the last thing to consider is a high dosing regimen and the sampling time points, which are linked because it's really important to ensure that there is suitable washout period between the ADA time points. And when that's not possible, then there is a need for more high and target tolerance assays as well.
So Agilex Biolabs is really a multicultural environment. We have scientists from all over the world that all bring slightly different perspectives to assay development. And we have over 6 years of experience, combined experience in designing, optimizing, developing, and validating immunogenicity methods according to regulatory and safety guidance. We have expertise not only in standard bridging methods, but also in non-standard methods like Assays, SPEAD, and PandA. We're actually going to touch on those methods during our presentation today. And the use of those methods really allows us to continuously meet analytical challenges, including development of methods that are highly drug and target tolerant. And we offer immunogenicity assays for all large molecule drugs, including biosimilars, MAPs, biospecific antibodies, recombinant proteins, and peptides. So now moving on to the actual case studies, starting with case study number 1. So this case study, as part of this case study, Agilex was tasked to develop an ADA assays for humanized monoclonal antibody that for the purpose of this presentation going to call drug Z. And the clinical study design was such that we were expected to develop a method that has a drug tolerance of 40 microgram per milliliter, which perhaps isn't a lot, but there was added complexity of a soluble target being present at quite high concentration of 2000 nanogram per milliliter. And the target was actually multimeric and was able to mimic the immune response and create false positive samples. And we've called that case study format screening approach because we've actually tested a high number of different formats. So our standard approach to developing and optimizing ADA assays is to always create a set of samples, including PCs, at least the PC at 100 nanogram per milliliter, drug tolerant samples, and also some individuals. And we've actually found that by using this set of samples in the very first optimization experiment gives us the best data to make conclusion and plan next steps. So the first format that we've tested was bridging with and without acid dissociation. And in this format, we've observed sufficient sensitivity but insufficient drug tolerance. The data is not presented here, but what I wanted to show you is how much the individual response varied between the tested matrices. On the graph to your left, you will notice that there was a huge scatter of those responses. But even after removing the clear outliers, the graph to your right, you will notice that that response is still very varied from 50 to 700 ECLs. And based on that, we basically concluded that it's likely that the presence of target is causing this scatter of results, and we've abandoned this format and moved to a next one. So the next format that was investigated was actually a non-standard method, affinity capture elution, known as Assays. And for this method, we went for a maximum allowable MRD of 1 in 100. And again, in this format, we've noticed that we achieved appropriate level of sensitivity, but again, insufficient drug tolerance. Referring you back to the graph, you will notice that with no drug, we've got a good response with the PC at 100 nanogram per milliliter, and as soon as little as 15 microgram per milliliter of drug is added, that response goes below the estimated cut point. And again, this format was put aside, and we move to the next one. The next format that was investigated was precipitation and acid dissociation, otherwise known as PandA. And we've actually introduced multiple alterations of the PandA method. I'm not going to discuss them in a lot of details, but basically we've focused on PEC buffer concentration, MRD, and capture and detection reagent concentration. But no matter of the the alteration, this format was unable to meet the sensitivity requirement. Then looking back at the graph with the red line representing estimated cut point, you will notice that only PC at perhaps 200 nanogram per milliliter would allow us to to see positive response. At this point, we left this particular format and moved to the next one. And the next non-standard method that we tested was solid phase extraction with acid dissociation, otherwise known as SPEAD. And what's interesting is that after very first optimization experiment, we actually achieved appropriate level of sensitivity and drug tolerance. But unfortunately, the assay suffered from target interference. And looking at the graph to your left, you will notice that presence of target at 2000 nanogram per milliliter was actually giving a response higher than the PC at 100 nanogram per milliliter. From there, we were really able to direct our focus on optimizing this particular method. And we've introduced a few anti-target reagents, including multiple antibodies and also receptor. And we've introduced them to the assay at different points. What I mean by that, they were included as part of different steps of the assay. Looking at the graph to your right, you will notice that receptor was actually the optimal molecule to conquer the target interference. To summarize the case study number one, we've obviously screened multiple formats, and that really allowed us to exclude certain methodologies straight away and meant we could dedicate our focus on a particular method. And by further refining this particular method, we were able to produce fit-for-purpose assay that met the requirements for the drug and target tolerance. And one final note from myself is this graph to your right represents how much the individual responses actually varied from format to format. And you will notice that the optimized SPEAD showed quite tight, nice responses. You could argue that PandA is the same, but I think that this is only a reflection of lack of sensitivity in this particular assay format and not necessarily uniformity of response. That's it from me, and I will now hand over to Emily for case study number 2.
Thanks, Agata. I just want to interrupt there. It was a really great case study to show how we can rapidly screen formats and reduce our clients' timelines. But just as a question, what kind of impact would, when you see that type of variability in individuals, would it have on the overall assay development?
I think if your assay displays high variability in tested individuals, you should be questioning the assay. Perhaps matrix interference or more specifically target interference plays a role here. But overall, increased variability in individual matrices will overinflate the normalization factor, and with that, your assay will seem to be less sensitive and less drug tolerant. And opposite can be said for instances where that biological variability is very low. In those instances, you will see an assay being extremely sensitive and extremely drug tolerant.
Great, thank you.
Thank you both. So now let's look at the case study number 2. So the drug was also a humanized monoclonal antibody. The study requirements were that we needed an assay that was able to tolerate up to 20 microgram per mil of drug. There was also the presence of soluble target in the samples, and so the assay needed to tolerate up to 200 microgram per mil of target. And one of the most challenging aspects for this project was that the drug was designed to have a very high affinity for its target. So we started with the standard homogeneous bridging assay, but this assay format showed both drug and especially target interference. So this created some false positive and false negative results, like Morgan explained briefly. So then we took a format screening approach. Like Agatha mentioned, we tested ACE, SPEAD, PandA, more complex methods. But these were not suitable. So we also investigated the use of anti-target antibodies and also heat treatment to try and denature the target. However, all of these formats showed that there was either a lack of sensitivity or it did not remove the interference sufficiently. So what we did was we went back to the homogeneous bridging format and we did some acid buffer scouting. So the acid that was used originally was a 0.3 molar acetic acid, pH 3. And we compared it to 5 other different acids, and we looked at sensitivity and drug tolerance. So here on the left, we can see the LPC was set at 100 nanogram per mil, and it was also spiked with 20 microgram per mil of drug. All of these conditions gave sufficient sensitivity and drug tolerance, but you can see there was a slight improvement with the use of the 0.1 molar HCl pH 1.5 and both the glycine buffers. So after that, we looked at the target interference. So we used a panel of individuals and we spiked them with 200 microgram per mil of target. And then we treated them with the same acids. And here it was pretty clear that the only acid that removed the target interference sufficiently was the 0.1 molar HCl pH 1.5, which is presented in black on this graph. So in summary, this assay with the optimized condition was sent to validation. It demonstrated a sensitivity below 10 nanogram per mil, a drug tolerance of 75 microgram per mil, which was more than what we needed. And it was able to tolerate up to 200 microgram per mil of target. This project has showed that the drug and target complexes can be quite challenging, especially when they have high affinity, because the use of anti-target antibodies is not always applicable. But what we also saw is that the use of more complex analytical solutions is not always the best approach. Here, for instance, the homogeneous was what gave us the most suitable assay. And that was only because the key to develop a suitable assay was to have to find the optimal pH to dissociate the drug and its target. Let's move on to case study 3.
Before we move to case study number 3, can I ask you, Emmeline, to expand on what are the benefits of using such a straightforward method?
Yes, of course. So I guess, first of all, it's quite easy to perform. So for an analytical point of view, it's much better for the analyst. It also shows that there's usually improved assay robustness. It also means that it takes less time to perform in the lab, which means that there is a time to result significantly reduced, and therefore there is a faster turnaround for sample analysis. Morgan, do you want to add anything?
Sure, yeah, I think you're absolutely right that really these simple methods improve the speed to result, which is always beneficial for our clients. I think on top of that, it's also important to consider that these simple methods are kind of the most robust when it comes to detecting all the available isotypes within a clinical sample. they have minimal wash steps and are quite capable of detecting a lot of different antibodies. So it's always something to bear in mind that if your trial requirements are to detect everything possible, that sometimes this most straightforward is the best answer.
Thank you.
Thank you.
So moving into case study 3. So here we've again looked at a humanized monoclonal antibody therapeutic, but what was quite interesting here was that we actually had no target tolerance issues within this specific case study. And this was because it was a non-soluble target, meaning that there would be no target present in the patient sera samples. Where the real challenge came from was the high requirements of drug tolerance in the assay. In this instance, needing up to 500 microgram per mil of drug tolerance. So the first thing we wanted to do was quickly screen those formats, just as Agata presented in her case study. So here we screened the ACE, SPEAD, and PandA formats. And this is what this graph is showing, is that really all 3 formats gave adequate sensitivity, some better than others, although even the PandA format represented in pink was still giving a signal to noise of around 2 to 3 in this first instance. What was really telling though was as soon as we put any free drug into those positive control samples, the signal in all 3 assay formats was just completely wiped out, meaning that we weren't gaining drug tolerance that we required for this particular clinical trial. So what we wanted to do was go back to the literature and really go back to kind of first principle thinking. And this seminal paper by Jogby et al. Was really the first paper around the PandA format. And within that, they were routinely seeing drug tolerance levels similar to those that we required for our assay. So just looking at the methodology of the PandA format, it's really based on 2 things. So saturating your anti-drug antibodies with a high amount of free drug, and then using polyethylene glycol to pull those antibodies out of the complex matrices. After that, there's an acid dissociation step, which then frees all of those ADAs again, and then a subsequent detection. So leaning into Emmeline's case study, we thought that acid optimization would be a first great step to see if we could improve the signal generation within the PandA format. So the 0.3 molar acetic acid is actually the standard acid used in this assay and is the recommended acid from the first paper. And you can see it gave us quite good signal generation across a range of PC concentrations. What was interesting was as soon as we tried other acids in this format, it completely wiped out the signal, indicating either that it wasn't breaking apart those complexes that we'd formed, or subsequently it had maybe destroyed the ADAs present in the samples. So we moved ahead with the 0.3 molar acetic acid, but again, we wanted to go back to the literature to see if there was any alternative thinking that we could come up with on how to optimize this assay. So we went right back to 1978 in some of this literature search, and we found really that polyethylene glycol precipitation isn't a new approach to getting proteins of interest out of complex matrices, but it is quite novel in the bioanalytical world. So we went back to some further manufacturing literature, and we found that often they were actually optimizing their polyethylene glycol precipitation by changing the pH of the buffer they were using to precipitate more optimally the protein they were looking for. So we wanted to apply this thinking to our assay. So what we did was we exactly that. We took our PEG buffer, our polyethylene glycol buffers, and we pH adjusted those buffers. And we wanted to do this by surrounding the isoelectric point of the drug to give us a kind of known pH to target. So the isoelectric point was around 8.5 for our drug. So we spanned that range, which you can see on the left-hand graph here. What was really interesting was when our pH buffer formulation of PEG sat just below the isoelectric point, we did see a slight increase in the sensitivity of the assay, as you can see that kind of lilac colored line just kind of shifting higher than the others. Next, to make sure that this was having a kind of relation then to an improvement in the drug tolerance, we wanted to kind of compete it head to head with the original buffer formulation. So in the right-hand graph here, you can see our low positive control at 100 nanogram per mil also spiked with differing levels of free drug in that head-to-head comparison. And you can see that we did gain a slight increase in sensitivity, our signal to noise ratio just sitting below 2 for our LPC, which in this instance was well above the estimated cut point of the assay. What was really good to see though was that when we did have free drug in there, by optimizing that pH of the buffer, we had managed to increase the sensitivity within those drug tolerance samples, meaning that we were much more robustly capable of detecting our anti-drug antibodies when there was free drug present. So in summary for this case study, as we've kind of discussed throughout the 3 case studies today, drug tolerance is always a major hurdle for immunogenicity. And actually, in this instance, we've kind of demonstrated that even the most tolerant formats in the literature still require some further optimization for a specific drug of interest. And what we really wanted to drive here was for bioanalytical scientists to truly think like a scientist and draw on their own experience, as well as the experience of maybe other industries who have a vast wealth of knowledge to improve their bioanalytical assays. In this instance, obviously, us going back to manufacturing really helped to create a fit-for-purpose assay for our end client.
Thank you, Morgan. I just had a quick question. What else do you need to consider when using more complex methods?
That's a great question. Thank you, Emmeline. I think to use the PandA format as an example, where we are relying on such a heavy use of precipitation, it can be quite important to consider what's actually happening in terms of patient samples. So we tend to develop these assays using a positive control. But actually, when you can look at this in a PandA assay, when you compare a precipitated sample to a non-precipitated sample in a recovery experiment, sometimes you can actually see quite a big difference between the signal generation of those 2 variables. So sometimes it can be quite important to actually have multiple positive controls, maybe of a range of affinities, so that we can make sure that we're actually adequately characterizing the assay for the range of responses that we might see in our patient sera, and make sure that we are robustly detecting our anti-drug antibodies. I don't know if anyone else would have anything to add to that.
Well, I think that's a great answer, Morgan. And perhaps one thing which I would add is that we would also often have data from a standard bridging method without acid that will really give us the baseline of the sensitivity of the method.
Great, thank you. So to conclude today's presentation, really what we've been trying to talk about is how to develop a fit-for-purpose assay that meets both the assay and clinical requirements of any given clinical trial. Although there were some historical challenges of reagent availability and technology choices, really these have now been superseded by complex modalities and high dosing regimens. And because of that, we need to be relying on our scientific knowledge and really letting the science lead to produce the most fit-for-purpose assays. By doing this, we actually increase the quality and the robustness of the assay, allowing it to move through the validation phase much easier and be deployed to sample analysis with minimal repeats and issues coming up in the assay, therefore reducing, again, that time to result. What this ultimately means for our end clients is that they actually have a significant cost saving by spending just a little bit more in that development time and making sure that that assay is the most appropriate it can be. So thank you for listening today. And I'd just like to say a few acknowledgements to both Emmeline and Agatha as my co-presenters, but also the wider team here at Agilex. So Kurt Sales is our Chief Scientific Officer for providing approval for us to do these case studies. Mark Fiorucci as our Director of Immunoassays, again, for providing his support and permission to conduct these case studies. And finally, to an external vendor, BioRender, for allowing us to use their platform to create some of the pictures in today's presentation. So with that, I'd like to move to the questions and answers section. And again, if you have any questions for myself or any of the other panelists, please do submit them.
Well, thank you so much, guys, for such a great presentation today. I do have some questions coming in from the audience. Let's go to the first one. As modalities evolve, what unique challenges are faced by bioanalytical scientists when considering immunogenicity assays? Who would like to take this one?
Thanks, Janky. That's a great question, and I'm happy to take that one. So I think over the last kind of decade, we've seen a real diversification in modalities, kind of shifting away from standard monoclonal antibodies through to antibody conjugates, bispecific antibodies, as well as cell and gene therapies. And I guess to use the cell and gene therapies as an example, really here, these have created some really quite unique challenges in the terms of immunogenicity. So to use a gene therapy as my first example, typically these are using a viral vector to actually deliver the payload into the patient. What can be a challenge here is that many of those viral vectors actually have an immune response already there in the population that you're actually trying to dose. So it can be quite difficult to discern what's kind of a baseline response to those viral vectors and what is a response that's actually been induced by the dosing of that drug to the patient. Likewise, when we think of cell therapies, so to use CAR T cell therapies as an example, we really need to start thinking about using new technologies such as flow cytometry so that we can discern what response is being driven against the therapeutic cells as opposed to the wild type cells. So really, it's a case of being flexible as a bioanalytical scientist, and as these new modalities are coming in, really, again, thinking outside the box to make sure that we can meet all of our clients' requirements and make sure that we're delivering a fit-for-purpose assay.
Excellent. Thanks for that, Morgan. To have another question coming in. You have mentioned here some non-standard ADA methods like Assays, PandA, and SPEAD. What other methods can you try?
Thanks, Janki. I can take that one on. So I believe we've covered majority of non-standard methods. However, at Agilex Biolabs, we also have experience in merging the methods together and creating hybrid. By that, I mean merging Assays with bridge or SPEAD with bridge. And we will do it by taking the Assays and SPEAD assay to the second acidification stage and then transferring the acidified samples into a standard bridging method. And we found that this really is a powerful assay to push that drug tolerance or target tolerance even further. And in addition to that, you've got an option of introducing use of magnetic beads or replace acid with high ionic strength dissociation. Maybe it's an overstatement to say that possibilities are endless, but you truly can, always tweak and change certain aspects of the method to make sure that you're developing a fit-for-purpose assay.
Okay, great. Thank you for answering that, Agata. Have another one here. If all assay formats fail to meet the study requirements, what can be done to ensure a positive clinical outcome?
Thank you, Janki. I guess I can answer this question. I guess without repeating what Agata just said, there are pretty much an endless list of parameters that can be optimized, different formats. So before saying that everything fails, there are options, but it's not always realistic, especially due to time constraint or cost. So what's important is to have a very open communication with the sponsor to make sure that they are aware of the assay limitations. Sometimes it can be valuable to redesign the clinical protocol to make sure there's enough washout period between the time points if the drug tolerance is an issue, for instance. But then again, if that's not possible, it's really a matter of having that open communication and making sure that they understand what their data is telling them. If they need to miss some time point, it's all about the conversation with them. So the sponsor is aware. Thank you.
Great. Thank you for that. Well, that's all the time we have for today. So thank you to everyone in the audience for tuning in, and thanks to our panel for today for sharing their time and expertise with us. If you would like to rewatch the webinar or share it with your colleagues, a link for on-demand viewing will be sent tomorrow. I'm Janki Patel with today's sponsor, Agilex Biolabs. Thank you all for joining us, and thank you again to Endpoint News for hosting us today. And we look forward to seeing you at the future webinars. Have a good day.